Okay, this is the experiment of clinical biochemistry practical, and the aim of the experiment is estimation of serum triglycerides. So we require some of the requirements while performing this test in the lab. What requirements do we need to procure? So let's know about this. Equipments and apparatus required: colorimeter, centrifuge, autopipette, test tubes. So these are the equipments and the apparatus we need to procure first. There should be colorimeter available. There should be centrifuge, and you must know the use of this instrument. The colorimeter is used for taking absorbance of the reaction mixer. Okay, if the color is more, then absorbance will be higher, and if the color is less, then what happens? There the what we say, the absorbance will be less. You can mute yourself, right? Because it may can create some noise from the background. We are studying the experiment estimation of triglycerides in serum, and the requirements we need to procure are the colorimeter and the centrifuge. The role of centrifuge is to separate serum from the blood sample. The autopipettes will help you to pipette a specific volume of reagents and sample, and the test tube is for mixing the sample and the reagents. What chemicals do you need during the procedure? So we require triglycerides kit. So it's a ready-made kits are available where we have different enzymes in the reagents, and we require distilled water or blank. Apart from this equipment and chemical, we need to arrange sample, patient sample. So this is the instruction for procuring sample. Use unhemolytic serum or plasma. So you have to collect blood sample in a plain vial so that you can separate serum. Okay, and ensure that the serum is not hemolyzed. Okay, what is non unhemolytic serum? It means the red cells are not lysed. Okay, the serum color is straw color. Okay, light yellow color, or because of the bilirubin, small amount of bilirubin present in the serum, and it should not be red color, right? The serum should be straw color or light yellow color. And fasting for 9 to 12 hours before the triglyceride test is required. So you need to give the instruction to the patient that uh, there should be the fasting state of 9 to 12 hours. So the better time to do the test is in the morning. The patient should do the normal dinner, and after overnight of sleep and rest, uh, the patient should report in the morning in the lab, and the patient should give the sample, right? General information about the test. Let's know about this. Triglycerides are the most common form of fat in our body. Okay, so human body has the fat, okay, the lipid, and the most common form of lipid is triglycerides. The triglyceride molecule contains three fatty acid and one glycerol. So when you study the chemical structure of triglyceride, you'll be able to see there are three fatty acids. So because of that, we say tri means three, and glycerides means it's a ester of fatty acid with the glycerol. Okay, so there are three fatty acid and one glycerol in the structure of triglycerides. High level of triglycerides in blood may raise the risk of coronary artery disease. Coronary artery is a artery that supplies blood to the heart. Okay, so there can be the risk of disease of coronary artery, and if there is a risk of coronary artery, when it is increased because of elevated level of triglycerides in the blood, then what happens? The coronary artery disease may leads to the heart disease or heart attack. Okay. Generally, a person's triglyceride levels are measured along with cholesterol test. So, when a doctor write or a test, triglyceride test, the doctor also write, the cholesterol test along with the triglycerides. Okay. So, person's triglyceride levels are always measured along with the cholesterol test. Normal triglycerides are below 150 mg per dl. So this is the normal range. That the healthy person triglyceride level should be controlled tightly below the 150 mg per dl. Okay, it should not exceed this level. Levels above 200 mg per dl are considered as a high levels, okay, or the severe level. Okay, and what are the factors that can raise triglyceride level? So we need to consider this, that what can increase the level of triglyceride level. So being overweight means you have the uh, more weight, body weight, and that body weight can increases the triglyceride level in the blood. Physical inactivity, a person who are is in the stage of sedentary lifestyle, do not do any physical activity, keep on sitting or lying on the bed most of the time, so it will increases the triglyceride level in the blood. Smoking is another factor. Smoking can stimulate the body to produce, to retain or to store or to increase triglyceride level in the blood. Excess alcohol use. So alcoholic person might have a more triglyceride level in the blood. A diet very high in carbohydrates. So a person who are taking a diet high in carbohydrate or even high in lipids, so their triglyceride levels are always elevated in their blood. Certain diseases and medications can increase triglyceride level in the blood, like diabetes. There may be the chances that the triglyceride level can be higher side, or if there is some medication taken by the in the patient, so that medication can also have a stimulatory effect to increase triglyceride in the blood. Some genetic factor disorder. Some genetic disorder means the inherited, the parental side, the maternal side, because of there's some gene which are abnormal gene, and some abnormal genes are inherited from the parents. So that genetic disorders can be responsible for the elevated level of triglyceride in the blood. Let's know the principle of the test. The test we are doing here is a enzymatic test. So this is the principle behind doing this test. So triglycerides here we have represented the principle here in the form of equation, chemical equation, enzymatic reactions, right? So you can see the triglycerides. If the sample contain triglycerides, then the lipase enzyme present in the reagent can break the triglycerides into glycerol and fatty acids. Okay, so this is the first reactions which can occur into the with the help of the reagents. Okay, the sample which contain triglycerides. Now the lipase present in the reagents will hydrolyze triglycerides in the sample and can produce a product glycerol and free fatty acid. Okay. Now the product of the first reaction is glycerol. Okay, can you see the glycerol? Yes. So this glycerol will consume in the second reaction. So glycerol in the presence of ATP, ATP means adenosine triphosphate. In the presence of glycerol kinase enzyme, we get glycerol 3-phosphate and 1 adenosine diphosphate. So this is the product of the second reaction. The glycerol which is produced in the first reaction will participate in the second reaction, and here it in the presence of the glycerol in the presence of ATP becomes glycerol 3-phosphate plus ADP. This reaction is catalyzed by GK enzyme, G for glycerol, K for kinase. Now what we see in the second reaction, we have what product? We have glycerol 3-phosphate we have got as a product, which will participate into the third reaction. So glycerol 3-phosphate in the presence of oxygen, so this is oxidized by to dihydroxyacetone phosphate, DAP, D for dihydroxy, A for acetone, P for phosphate. So this glycerol 3-phosphate is oxidized to DAP and S2O2. Okay, and this reaction is catalyzed by glycerol phosphate oxidase, GPO. Glycerol phosphate oxidase oxidizes glycerol 3-phosphate to DAP. DAP stand for, again I am saying, D for dihydroxy, A for acetone, P for phosphate. And we get H2O2, hydrogen peroxide. Now the hydrogen peroxide H2O2, which is produced in the third reaction, will participate in the fourth reaction. Okay, so H2O2 plus 4 amino antipyrine, this is 4AA, 4 amino antipyrine plus 4 chlorophenol. Okay, now complex of this, we get a quinone imine dye. Okay, and the color of the dye is, it produces a color. This dye produces, this complex quinone imine produces a color. So here what we can understand that more triglycerides means more quinone imine dye complex formation. So more triglycerides, more color formation, and more color means now that color we can measure the intensity of the color with the help of colorimeter. So here we can say the color is directly proportional to the absorbance, and absorbance is directly proportional to the concentration. Okay. This is what we have. I have written this the reactions what you have studied in the previous slide, that glycerol triglycerides are hydrolyzed to glycerol and fatty acid by lipase. What happens in the first reaction, that we get glycerol and fatty acid by lipase. Second reaction is the glycerol is then phosphorylated by glycerol kinase to glycerol 3 phosphate in the presence of ATP. So here we get glycerol 3 phosphate by glycerol kinase. And the third, the glycerol phosphate oxidase GPO, which oxidizes glycerol 3 phosphate to dihydroxyacetone phosphate DAP and hydrogen peroxide. Now the hydrogen peroxide react with 4 chlorophenol and 4 aminophenazone or 4 amino antipyrine, what we say, in the presence of peroxidase enzyme. And what we see that there is a quinone imine complex formation. The intensity of the color complex is measured at 530 nanometer, and the absorbance is directly proportional to the triglyceride concentration of the sample. But we understand here that if there is a high absorbance, at that time there will be the high level of triglyceride in the given sample. Now, I hope that you have understand the principle. Now let's focus on the procedure. How can we do the procedure? Procedure. What are the steps in the procedures? Let's know about this. Here, take 3 clean and dry test tube. Okay, you have to take 3 clean and dry test tube, and you have to label them as a T, S, B respectively. So all 3 test tube you have to label them as a T for test, S for standard, B for blank. Okay, and then after that pipette the respective ingredients in the respective test tubes. Now here this is the table. You can see we have a test here, standard and the blank, and these are the ingredients you have to add one by one into the test, standard and blank. Okay, so working reagent we have to add 1 ml in each test tube. Serum sample you have to add 0.01 ml in test label tube, and the triglyceride standard, which is having the concentration of 200 ml per day. So you have to add in the standard tube how much? 0.01 ml, and distilled water you need to add is 0.01 ml in the blank. Okay, so after adding this ingredient into the T, S, and B, means test, standard, blank tubes. Then after that you need to mix it properly, and you have to incubate at 37 degrees Celsius for 10 minutes. So incubation is very, very important, because at that incubation period at 37 degrees Celsius, what happens? The reactions takes place into the specific test tubes. Okay, because of the enzymatic reactions, at the end we get a quinone imine complex, and it produces a color, and the color intensity can be measured with the help of colorimeter. To measure the absorbance at 505 nanometer. So with the help of colorimeter, we can measure the absorbance of the test and absorbance of the standard at 505 nanometer against the blank. Blank is taken to calibrate the colorimeter. Okay, we can make it 0, and against the blank we can take the OD of or absorbance of test and absorbance of standard. Now, when we have the absorbance of test and absorbance of standard, now what we can do, we can do calculation. The formula is serum triglyceride in mg per dl is equal to OD of test divided by OD of standard multiplied by 200. 200 is a standard concentration for triglycerides. Now you can write the results, okay, by putting the value of OD of test, that is absorbance of test, and OD of standard means absorbance of standard. So you can put the value in this formula and you can multiply by 200 and you can get the results. Now you can write the results in serum triglycerides in it's mg per deal, right? It's mg per deal. Okay, so always write the value in mg per dl, and then the normal range, that is what the serum triglycerides level is always below 150 or 160 mg per dl. Okay, should be less than 160 mg per dl. What is the significance of high triglycerides? If the value is above the normal range, what it is called as a high triglyceride level. Okay, so high triglyceride level may result in hardening of arteries. When the person has a high level of high level of arteries, sorry, high level of triglycerides in the blood, so it can result in the hardening of the arteries, and that process we call them as atherosclerosis. Atherosclerosis is defined as a hardening of arteries, okay, in simple language. And this will increase the risk of stroke, heart attack, and heart diseases. Okay, so these are the clinical significance that triglyceride level should not be a higher level in our blood. Otherwise, it can increase the risk of the heart disease or the stroke-like conditions, right? And the error analysis we can do, that there can be the chances of errors. So ecteric and lipemic sera may interfere with the test. What is ecteric? If there is a the serum from the jaundice patient, so their serum is yellow color, dark yellow color or brown color. Okay, so in that condition, it may can interfere with the absorbance. Similarly, if you take the serum from the lipemic sera, lipemic blood, means if the patient has taken the lipid diet, and there is a lot of lipid in the sera, so automatically it can increase the level of the triglyceride level. So ensure that the syrup should not have the lipids containing milky sera. Okay, usually this appears when the person, when you collect the sample just after half an hour of the meal. Okay, so the dietary lipids may can mix into the blood, and there can be the lipemic sera. Okay, you can, so avoid collecting sample in the in the daytime. Ask the patient to come in the morning time in the lab, and the sample should be collected in fasting condition. And ensure a complete clot formation has taken place prior to centrifugation. So when we do centrifugation process, so to separate the serum, so first of all, allow the blood to clot properly, and once the blood has clotted properly, after that you can do the centrifugation, and after that you can separate the serum from the top of uh, the blood sample. Okay, so we have completed. We have come to the end of the slide. Now we can discuss if you have any questions. So we are open for question and discussion now.